All right, this is Microbiology Lecture 8, and it's on microbial genetics, and I would hope that some of it, at least, is a review rather than the first time you've seen it. Now, you may have forgotten parts of it, certainly, but I hope that nothing here is 100% new to you. First, let's start with some definitions of terms that you might want to go back on. If I use one of these terms, you might want to check the definitions of them. Genetics is the study of genes and their function. A gene is a segment of the DNA which codes for a functional product, and that normally would be a protein. The genome is all of the genetic material in a cell, and if it's a multicellular organism, each cell contains the whole genome, which would code for everything needed, all the information required for that particular organism to exist. Genomics is the molecular study of the genome, or genomes. The genotype is the genetic makeup of an organism, and the phenotype is how those genes are expressed. Sometimes they are silent, they're not expressed, because there's always two copies in an organism, and one copy of the gene may be dominant to the other. So in the end, the phenotype is not necessarily does not necessarily directly one-to-one reflect the genotype. There may be genes in there certainly that are not expressed. So the phenotype is actual expression, the genotype is the genetic makeup. Now, even a simple little prokaryotic organism, a bacteria like E. coli, has chromosomes. It has one chromosome and it's circular, unlike eukaryotic cells that have linear chromosomes. There are a lot of genes, doesn't matter how many, but there are a lot. Again, not anywhere near as many as you would find in a human, for example. There are many base pairs, AT and GC, which those are base pairs. AT is a base pair, GC is a base pair, which are in those genes in the genome of the bacteria. They may not be as complex as humans or mammalian organisms, but mammalian organisms, on the other hand, are relative to plants fairly simple. Many plants have a much larger genome than mammals do. Okay, now there's certain directions in which information flows from the genome. The information in the genome is used to code for production of new cells or new proteins. So within a cell itself, where we're looking at a single cell, a single generation of cells, if you look at that cell, it has a genome, which is the chromosomal DNA, and that can be expressed in that cell, and it needs to be expressed in order for the cell to remain viable and to have the ability to carry out metabolism, biochemical reactions, and to grow. So all the information is in the DNA, in the genome. There are genes in that DNA, and when a gene is activated, it is transcribed. So transcription is the information in the DNA, in a gene in the DNA, is being used to generate a molecule of RNA. And usually that would be a messenger RNA molecule. And then the messenger RNA molecule is used, the information in there, which is originally from the gene, is used to synthesize a protein. And so that is translation. So proteins are synthesized from the information in the messenger RNA transcript, and that occurs at the ribosome, and a protein is produced by translation. So we go from DNA to RNA, messenger RNA, that's a transcription, and you get a product is a messenger RNA transcript, which has the information that was in the gene and the DNA, and then that information is used to carry out translation. The information is used to make a protein from that messenger RNA information. And so that is translation. The final product is a protein. So that's what occurs within cells. At the same time, you have a different kind of flow of genetic information, and that is when a cell divides. And for a cell to divide, you have to have replication of the DNA. So DNA replication occurs. You have twice as much DNA, and everything else replicates itself. You get more of everything that makes up the cell, and the cell divides by, in the case of bacteria, by binary fission. In the case of eukaryotic cells, by mitosis, and you end up with two daughter cells that have all of the same identical genetic information, each of them, as was in the parent. So that's DNA replication. Now, there is a possibility of information being shared or shifted over from one organism to another, and from one cell to another, and that's called recombination. Here, recombination, the product would be a recombinant cell, but we use the term mainly recombination. And so you have some of this material here in this cell. I'm going to say a piece here that is transferred over and inserted here. So that's a recombinant cell. It has its own information plus information from another bacterial cell. Okay. And these are the different ways that information can flow within a cell in the same generation, either because the cell uses genes, or it can happen that from one generation to the next, and by generation I mean going from parent cell to daughter cells. So DNA replication occurs, and the cell divides into two new daughter cells. This is DNA replication. Okay, in terms of the structure of DNA, and I again would hope that this is review rather than the first time you saw it. The building blocks, of course, I know it is review. The building blocks, of course, the individual monomers of the polymer are nucleotides. In the case of DNA, you have two strands. They're arranged in a double helix around each other. Maybe I can make that in a different color, the other strand, just so you can clearly see it is a different strand. It is different, but it is complementary. They are complementary to each other. Complementary meaning that if there's an A on one strand, will always be a T on the other. T here, A over there. And the other type of base pairs, AT is a base pair, and the other is GC, right here, GC. Okay, if there's a G on one strand, there'll be a C on the other one. So the four nitrogen-containing bases that can occur in DNA nucleotides are adenine A, thymine T, cytosine C, and guanine G. And they will base pair with each other. Where if there's an A on one strand, there'll be a T on the other. If there's a G on one strand, a C on one strand, or a G, there'll be a G on the other, or C, ATGC. The backbone, it's referred to as a backbone, refers to not the nitrogen bases, which are like ladders, steps across, but refers to the second and third part of the nucleotide, which is the deoxyribose, a carbohydrate group, and a phosphate. And here you see that there's the deoxyribose, there's the phosphate, there's another deoxyribose and a phosphate, and it's called the backbone. And that's what really forms the polymer, with these nucleotide, with these nitrogen-containing bases sticking out across in this way. And these two strands, this strand here will, this strand here will form hydrogen bonding with a complementary strand here, which has T's or G's or C's or A's wherever there's a the appropriate complementary strand sequence on the other side. So wherever there was a T in one, an A in one, you'll have a T on the other, C, G, G, C. That's the structure that it looks like. The strands are held together by hydrogen bonding, hydrogen bonds. There will be always 2 hydrogen bonds between AT base pairs and 3 hydrogen bonds between GC base pairs. And the strands are arranged, the 2 parallel strands are arranged in what is called an anti-parallel conformation. They are anti-parallel always. And what that means is that you have always a 5' end and a 3' end for each strand. So there's a 5' end and a 3' end. And what that refers to, the number and the prime refers to which carbon we're talking about. You see, each of these deoxyribose molecules has 5 carbons. They are pentose sugars. So they are not pentamers, they are pentose sugars, 5 carbon sugars or carbohydrates that in which each carbon has a particular position, 1, 2, 3, 4, 5. If the last phosphate is attached to the 5 carbon, if this is the very end of it, then this will be the 5' end of this polymer strand here. And as you can see, this here, right there, I'll do it as an arrow, right here in that position, that's the 3' carbon, so that's the 3' end of the molecule. Now this strand is going 5' to 3' in this direction, and what is meant by anti-parallel is that the other strand will be going in the opposite direction. It'll be arranged in the opposite direction, and so you will have a 3' end here and a 5' end there. So you always know in this kind of base pairing situation, if you know one end, that this, for example, is the 5' end, then you will know what this end is, and you will know what the end here on the other strand is, and you'll know this just by knowing this one single end is 5'. Similarly, if you know that this is 3', then this has to be 5', and if this is 5', this must be 3, and if that's 3, this must be 5, etc. This next slide shows a segment of DNA which is undergoing replication. So it's looking just at a short region or short segment of the DNA, and this part is undergoing replication, where the 2 parental strands are shown here and here, are unwinding at 1, and a new daughter strand complementary to the parental strand is made on either side. So there's a new daughter strand that's being made here and another one that's being made here. Now I just want to point out to you that DNA strands, again, are anti-parallel, meaning that if you know that this end is 3', then this end must be 5', and if this end on this strand is 5', then this end must be 3'. And since new strands are always made in the 5' to 3' direction, then you can understand that on this side where this new daughter strand is being formed, and remember it's formed as a complementary strand to the parental strand, so wherever there's a T on the parental strand, there will be an A put in on the daughter strand. I'll make that darker. Wherever there's a T on the parental strand, there'll be an A put in on the new daughter strand as it grows. Wherever there's an A on the parent, there'll be a T coming in on the daughter cell, on the daughter strand. G, C, T, A. So it's always ATGC base pairing. So the new complementary strands, nucleotides being added, are always complementary to the one facing them on the parental strand. And it's the parental strand that is the template for addition of new nucleotides on the growing daughter strand. I point out to you, and again remind you, that new strands are always made in the 5' to 3' direction. And what that means is that this new daughter strand here, this one, will grow by addition of new nucleotides in this direction. So new nucleotides are added on going this way in this direction. Now, which is fine because the replication fork is unwinding and it's, exposing more and more template, the parental strand template, and another new one will come in. Say, for example, if this nucleotide here is a G, then a new nucleotide will be added here as a C. If this one is a T, then a new one will come in and be added as an A, etc. And that is in the 5' to 3' direction of the growing daughter strand. Now the problem, or seeming problem, occurs on the other parental strand, which as the parental DNA is unwinding at this point, the new strand still must be on that side. This is the 3' end, and this is the 5' end of the parental strand. And new strands have to be made in the 5' to 3' direction. So that would be this way. This end is 5', and this end is, as you can see, as indicated, is 3'. Whoops. Let me just fix that. This end here is 3'. I know that because this side is 5'. So this is 5' up here and 3' down here. And the direction of synthesis must be always in the 5' to 3' direction. That means that the new strand has to be made in this direction. And as you can see, the new nucleotides are being added in that direction. Here is an A nucleotide being added right there because there's a T on the parental strand. There's a T being added after that because there's an A, and now the most recent one coming in is the C here because there's a G on the parental strand there. So it is made in that direction. A short segment is made, and then there's a pause and more parental strand unwinds, and then a new short segment of daughter strand DNA is again made in the same direction, 5' towards 3'. So essentially, if we look at this parental strand right here, which ends here, if I draw it in this fashion, okay, and if the replication fork is starting right there, as you get unwinding of the parental strand, a new strand of DNA is made like that, in that direction, 5' to 3'. And then another new one is made going back, and the replication fork keeps moving back up the parental strand as you get unwinding. So you get a continuous sequence of new daughter strands being made in the direction that I draw each arrow. You see? So they're always being made 5' to 3', but they're made in little short segments, and those short segments are called Okazaki fragments. Okazaki fragments. Okay? Okazaki. Okazaki is not with a J, it's with a Z. Okazaki. I wrote Okajaki. It's Okazaki. Okay. Let's move on. Now the enzyme that carries out that synthesis of the new daughter strand, on both sides it's the same enzyme, is called DNA polymerase. And again, I remind you that the new daughter strand is always made in the 5', 3' direction. If we have two parental strands, as shown before, and you have unwinding of that, and if this here, I'll draw it quickly, if this here is the 3' end of that parental strand, and that's the 5' end of that parental strand, then this will be 5' here on the parent, on that parent, and this will be 3' down here on this parent strand, and the new strands will always, remember, be made in the 5' to 3' direction, and so it'll be a new strand forming in this direction, 5' towards 3', right? And I'll put a 3 here, indicating that that is the new strand there is 3', and 5' to 3'. First, this one, as the parent was unwound, this new strand was made, and then the DNA polymerase moves back and again makes another Okazaki fragment in that direction like that, and then another one. In the end, what you get is this kind of addition during DNA replication of new nucleotides, new complementary nucleotides on the newly forming daughter strand. The energy required for the process comes from the cleavage or cutting of this phosphate bond right here. Let's do that in a different color. The phosphate bond right there is cleaved, and with cleavage of that bond there's energy released, and these two phosphates are released. They're shown here, and you get this phosphate. These are all phosphates. These round symbols here are all phosphates, and the deoxyribose carbohydrates are shown here. So you get this phosphate, deoxyribose, pentose carbohydrates there. That's the backbone, and you have newly forming strands being made. Always in the 5' to 3' direction. Here's another diagram showing that same idea as I showed earlier. The new strand that is being made continuously which is indicated here, this one. That's the new daughter strand that's being made in the 5' to 3' direction. That is called the leading strand. So that's the leading daughter strand. And the other one that's made discontinuously, like this, and then followed by this, followed by this, followed by this, always in the 5' to 3' direction. That's called the lagging strand. So the new daughter strand that's being formed discontinuously in Okazaki fragments is called the lagging strand. Now, and here you see the name Okazaki fragment. So all of these short little segments that were made in the 5' to 3' direction using of the newly forming lagging strand are called Okazaki fragments. And then in the end, they are all joined together through the action of an enzyme that links them. So and that enzyme is called DNA ligase. DNA ligase will join the discontinuous fragments of the lagging strand. Okay? Good. Let's go on. Now, DNA replication is called semi-conservative. The reason for that is that for each new copy that's being made, there will be in it one strand from the original parent and a new strand. And that you should have realized from what we were showing. We were showing that the leading strand of the new daughter DNA is made on the complementary to the one parental strand, and the lagging strand is complementary to the other, and so you end up with semi-conservative DNA replication. The other thing is that the other way that information is, that was DNA replication in terms of making a copy of the DNA, the genome, in order for the cell, so the cell can divide into two new daughter strands. But the other way that information is transferred is in a single bacterium or a single cell when it's metabolically active and is reading the information in the DNA to make messenger RNA from that. And that process is called transcription. And transcription involves the production of a messenger RNA transcript from the information in the DNA. And that messenger RNA will then be used at the ribosomes to make protein. The process of formation of the messenger RNA is by the action of an enzyme called RNA polymerase. So RNA polymerase binds to the DNA where the gene is and makes a messenger RNA complementary product or transcript. The product of transcription is called a transcript. Now, it's not only messenger RNA that can be made in this way, but also transfer RNA and ribosomal RNA. But we've only discussed up until now messenger RNA. Transcription of a gene begins when the enzyme, the RNA polymerase, binds to a promoter sequence that is at one end of the gene. And then at the 3' end of the gene, transcription will proceed in the 5' to 3' direction. And it stops when it reaches a terminator sequence or what's called a stop codon or terminator sequence. Okay, now transcription from DNA to messenger RNA is followed by the process of translation. So translation occurs at the ribosomes. In a eukaryotic cell, the ribosomes are in the rough endoplasmic reticulum or free-floating single ribosomes. In a prokaryotic cell, they are free-floating in the cytoplasm, and the messenger RNA will be the information messenger RNA is used by the ribosomes to form new protein, and that process is called translation. Now, translation is based on a genetic code, and the code is found in the messenger RNA, and it is present in what are called codons. Codons are 3 nucleotide sequences. I want to remind you that the nucleotides found in RNA are not ATGC that's present in DNA, but AUGC. These 4 nucleotides are the ones found in RNA. AUGC. The G complementarily can base pair with C, and the A can complementarily base pair with U. The DNA strand, which contains the genetic information, when it is transcribed to messenger RNA, what you get is that wherever there is a on the DNA side, and the newly formed messenger RNA is, I'll do it in a different color, the newly formed strand of messenger RNA is formed like that, always formed in the 5' to 3' direction. And again, so if you know that this strand is 5' to 3', this is going to be 3' to 5' for the DNA in the gene. So the new strand is always made in this direction, 5' to 3'. It always starts, and the messenger RNA, each three nucleotides is called a codon, and the first one will always be a start codon. So right here you have the start codon positioned there, and it will always be AUG. The codon sequence will be AUG. That is a start codon. AUG. So we're talking about the information messenger RNA, which was made on the basis of the DNA. So if you have an A in the messenger RNA, that's because there was a T in the DNA strand at that position, so that an A was put in. Did I say, yeah, T? Where there's a U here, that means there was an A. And where there's a G here, that means a C. Right? Messenger RNA does not contain T's, it contains U's. So TAC would be transcribed to AUG. And that is a start codon right there. So this is in sequences of 3. You look at it, the messenger RNA is viewed in sequences of 3, starting always with a start codon. And this is the table here. This table in this diagram explains what these codons code for in terms of amino acids that will be added into the newly formed protein that's synthesized on the basis of the information in the messenger RNA. So you may have, AUG as the first codon. You have to have it as the first codon because that's a start codon. And then maybe the next 3 will be GAC. And then the next 3 would be GCA. Notice that each of these codes for a particular protein. The abbreviated name for the proteins are shown. I'm going to do it in a different color. The abbreviated names in this table for proteins are shown here and here. You don't have to memorize any of it, but I would like you to know that AUG is coding for a start and also the amino acid methionine. So every newly formed protein is every newly formed polypeptide protein starts with a methionine. So here we have phenylalanine, leucine. These code for leucine. These code for isoleucine. There's methionine, valine, alanine. You don't need to memorize this at all. You would only hold it, perhaps, unlikely, you would hold it for a short period and immediately forget it. There's no point. There's absolutely no point. But you can look it up on a table like this. And I could ask you, if the messenger RNA has this following sequence, what is the protein sequence? Or I could ask you, if the DNA has a particular sequence, 3' to 5', then the messenger RNA has what sequence, 5' to 3', and what amino acids would that code for? The way this table is set up is that the first position nucleotide and first position nucleotides are all shown here in this position at the left there of the 3. The first position one is, in this case, U or C or A or G. And then the second one is shown across here. Second position is U, C, A, or G. And these are the third position nucleotides for the codons. And so if I said to you, what does GGU code for? Then you could easily look it up. The first, I said GGU, what does it code for? Well, the first letter or nucleotide, what I said, was a G. So it's going to be somewhere in this block here, right? And the second letter I said was G. So it's going to be clearly, has to be in this block. And then I said, I don't know what I said, let's say GGC, I think I said C. So then it has to be in the third position, a C, it's this one here. Is that clear? I hope so. So in this way, knowing a nucleotide, you can look up which amino acid it is coding for. The translation will proceed starting with the start codon and then keep going according to the 3 nucleotide long codons, bringing in a new appropriate amino acid to build up the newly forming protein polymer strand until it hits a codon that codes for a stop signal. And there are 3 possible stop signals: UAA, UAG, and UGA. UA, UAG, and UGA. Or UAA, UAG, and UGA. Those are 3 stop codons. The only one I need you to learn and memorize is AUG for start codon. The stop codons, if I asked a question about something involving that, I would give you the sequence of nucleotides for stop codons, but I do want to point out that none of these 3 code for an amino acid. It's just a stop signal. And it tells the ribosome to stop carrying out translation, to stop synthesizing and adding new amino acids to the newly forming protein strand. Let's take a brief pause. Okay, quite a bit of what I was saying is, in fact, written out on this slide, this next slide. We're looking here at the genomic information, which is in the DNA, is transcribed into messenger RNA. If a gene is turned on, it's transcribed to messenger RNA, and then that information is translated into a protein product. And messenger RNA, when it's translated, is translated in codons, and there are 3 nucleotides per codon. Translation of the messenger RNA always begins at the 5' end of the message. So the translation process is moving along, the ribosome is moving along from the 5' end of the messenger RNA molecule. It always starts at an AUG, and an AUG not only codes for a start codon, it's not only a start codon, but it is also the codon that codes for the amino acid methionine, which is abbreviated MET. And then the codons are read as the ribosome moves along the messenger RNA until it reaches a stop codon, which could be UAA, UAG, or UGA. Stop codons do not code for any type of amino acid. They are simply a stop signal. They do not code for amino acid. So, for example, if you're looking at a strand of messenger RNA that looks like this, then you should view it in terms of codons, so in sets of 3. And that's exactly what this is. It's the example here, separated into codons, 3 each. There are 7 codons, 3 nucleotides each. There are 7 of these codons here. They code for 6 amino acids, and a last one is a stop codon. So this would code for a 6 amino acid long peptide chain. Peptides are small proteins. And here, of course, is the table that shows the coding for translation of messenger RNA, AUG being the stop, and any of these 3 providing the start, and any of these 3 providing a stop signal. Now, when we look a little more carefully about what's happening during the process of translation, what you see is that you have a messenger RNA strand, and remember the translation process will proceed from the 5' end of the message towards the 3' end of the message. The process involves ribosomes, and a ribosome always has 2 subunits. So the messenger RNA, the ribosome attaches to the messenger RNA strand, and translation starts occurring with the AUG start codon, and of course that codes for methionine. Now the process is very carefully controlled. The information is in this messenger RNA that codes for, the information codes for a new protein. All right. And the new protein, of course, is made up of a specific sequence of amino acids. The sequence is determined by the sequence of codons in the messenger RNA. What allows for a particular amino acid to be brought in, and then another one, and then another one, is the fact that there are transfer RNA molecules, which on a portion of the transfer RNA molecule, you will have an anticodon. And the anticodon has a particular sequence. And notice here, this is the anticodon for methionine. AUG not only codes for, is not only a start signal, a start codon, but it also codes for the amino acid methionine. And transfer RNA molecules bring in the appropriate amino acids one at a time. The appropriateness of the tRNA is that there's a particular tRNA, a particular tRNA will bring in a particular amino acid. This one can attach and bring in methionine. It has the appropriate anticodon for that first codon. UAC can complementarily bind to AUG. The U will complementarily bind to the A to the U, and the U to this A, this A to this U, and this C to the G. That's why the process works, because of transfer RNAs. So the transfer RNA acts as a delivery system. So first methionine is brought in, and the next codon here is UUA. So an anticodon, a transfer RNA that has this anticodon on it will bring in this particular amino acid. AAU, shown here, is complementary to UUA, and so it will complementarily base pair, at least temporarily, bring in at the same time a leucine. LEU stands for leucine. The first amino acid has been added, now the second amino acid, and then the third amino, and they bind, a peptide bond is formed between them. Then the third amino acid comes in. It happens to be brought in by this transfer RNA molecule here, because this one has the anticodon CCA, which is complementary to GGU. And so it brings in glycine, and glycine will bind to the leucine. A peptide bond is formed to the leucine. So now we have methionine, leucine, and glycine. This in this example. And now this one is brought in, this phenylalanine. It's brought in by a transfer RNA molecule. This is the whole transfer RNA molecule with the amino acid. It has the anticodon AAA, which is complementary to the next three, and you should be able to tell me easily that this sequence here is UUU, because UUU, of course, is complementary to AAA. There you see that UUU that brought in the phenylalanine, was it? I believe. We'll have to check later, yeah. Methionine is coded by this. Then leucine is coded by that. Then glycine, then phenylalanine, and then another methionine. It's not a start. It's just a codon for methionine. AUG is not acting as a start codon here, because there was a start codon and there hasn't been a stop codon yet. So it's bringing in a methionine, and that methionine is, yeah, it's not that one, it's this one. This methionine here has just been brought in to bind to the phenylalanine, and the process continues on until all of the appropriate amino acids are added in the appropriate sequence determined by the message, each brought in by a different specific transfer RNA that has the appropriate anticodon, so it brings in the appropriate amino acid until it finally hits a stop codon, and here you see it, the UGA, and the process ends. The new protein is released. The messenger RNA actually can be read starting again. I'm sorry, the last tRNA is released and the ribosome comes apart according to this, but I can tell you that it's typically what actually happens is the messenger RNA is released and another ribosome will attach, and translation will continue until the messenger RNA is degraded. So in this way, we have the information in DNA being transferred to messenger RNA, and that's transferred to the protein by the processes of transcription and translation. I can tell you that what happens in prokaryotic organisms like bacteria is that transcription and translation are actually occurring simultaneously, and the translation is occurring very rapidly at the multiple ribosomes that attach to the messenger RNA. So here you see a DNA molecule. It's shown here. this RNA polymerase molecule is attached and making, I'm going to put it in green, is making the new messenger RNA strand, which is here. And polyribosomes are going to, yeah, that's the DNA. These RNA polymerase molecules shown here are moving along and making a strand of RNA, and the ribosomes will be making a new strand of protein from that information. It's not, protein's not shown there. Okay. Unlike prokaryotes, in eukaryotes, transcription and translation does not occur simultaneously. It occurs sequentially. First, transcription, and the transcript then undergoes a stage not occurring in prokaryotes. It undergoes processing before it gets translated. Remember that transcription in a eukaryotic organism, the production of a messenger RNA from the information in the gene, occurs in the nucleus because the DNA, of course, is in the nucleus. So if a gene in, say you have a strand of DNA, if a gene is, say, here in this location between those, so this area here is coding for a particular gene, then a message, if that gene is turned on, then a messenger molecule is made, messenger RNA. The new transcript, and of course messenger RNA is made by transcription, the new RNA transcript has to be processed first. In DNA in eukaryotic organisms, you have both introns and exons. Okay, these are regions of the DNA within the gene, and it is the exons that are coding. The introns are non-coding. So genes are made of exons and introns. The whole thing is transcribed to messenger RNA by RNA polymerase, as you've learned before, and you get a messenger RNA transcript, but it has to be processed because the introns are non-coding. Only the exon-derived RNA needs to be used eventually for translation to the protein. The intron-derived RNA has to be cut out. So the introns are cut out, the intron-derived RNA is cut out, and you get the mature messenger RNA transcript. Okay? And then that leaves the nucleus. All of this area in purple is the nucleus. Here they're showing that that's all these things occurring in the nucleus. Then the messenger RNA leaves the nucleus and goes into the cytoplasm to the ribosomes. So it has to undergo that processing, cutting out of intron-derived RNA. Message gets exported to the cytoplasm and goes to the ribosomes. Okay, I think that's enough for part 1 of Unit 8, and we'll stop here. That's the end of Unit 8, part 1.